Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University of Information Technology, Himachal Pradesh. Today I am going to talk about the module Neurotransmitters and Neuromuscular Junctions. This module is placed in paper Membrane Biophysics. Neuron is defined as specialized cell of nervous system required to transmit information throughout the body. Neuron cells generate action potential to communicate between brain and other body parts. Neurons interact with each other at the junctions called synapse. The presynaptic, that's a sending neuron, transmits signals to postsynaptic, that's a receiving neuron, to pass on action potential. Action potential in neurons gets propagated through the voltage-gated ion channels. The onset of an action potential at the axon terminus of a neuron causes the secretion of chemical moiety, which in turn bind to receptor on adjacent cells and cause changes in the membrane potential of these adjacent cells. The secreted chemical moiety is known as neurotransmitter. Present module primarily provides information of neurotransmitters, their structure-based classification, cause and mechanisms of their release and actions. Following learning objectives are prepared to achieve these information. Number 1. To elucidate the structural and functional characteristics of chemical moiety as neurotransmitter. Number 2. To classify the neurotransmitter on the basis of their structural features. Number 3. To classify the receptor of neurotransmitter on the basis of their mode of action. And lastly, number 4. To elucidate the mechanism of a neurotransmitter propagation through neuromuscular junctions. Synaptic transmission can be either electrical or chemical. Chemical transmission is more common, which involves neurotransmitters. Neurotransmitters or chemical messengers are endogenous chemicals that transfer signals across synapse from one neuron to another neuron or a target cell like muscle cell. Synaptic vesicles release neurotransmitters into synaptic cleft, and these neurotransmitters are received by the receptors on the target cell. A number of neurotransmitters are synthesized by amino acid precursor. Majority of neurotransmitters are of small size, approximately single amino acid. However, some neurotransmitters may acquire the size of large peptides. More than 100 chemicals have been identified as neurotransmitters. Neurotransmitters can be excitatory, that stimulate the brain, or inhibitory, that calm the brain and create balance. Inhibitory neurotransmitters easily get depleted when excitatory chemicals are overactive. There are following characteristic features which can make a chemical moiety as neurotransmitter. Number 1. The chemical must be synthesized in the neuron or otherwise be present in it. Number 2. In active state of neuron, the chemical must be released from it and create a response in a target cell. Number 3. A similar response must be generated during the in vitro experimental system on the target cell. And number 4. A removal mechanism must be present for the chemical from its site of activation after completion of the response. Additionally, the term neurotransmitter can be referred to the chemicals that carry the information between two neurons through influence on the postsynaptic membrane, or they have or little or no effect on the membrane potential. However, they can change the conformation of synapse. Number 3. They can communicate by sending reverse direction messages and alter the release or reuptake of transmitters. Now Coming to the mechanism of neurotransmitter releases. Neurotransmitters are stored in a synapse in synaptic vesicles, which are clustered beneath the membrane in the axon terminal, located at the presynaptic side of the synapse. The synaptic vesicles release neurotransmitter into the synaptic cleft by exocytosis after fusion with the axonal membrane. The exocytosis of neurotransmitter from synaptic vesicles involves the similar process of vesicle targeting and fusion events as you seen in intracellular protein trafficking of secreted and plasma membrane proteins. However, two features critical to synapse functions which are differ from other secretory pathways. Number one, the secretion occurs only at synapse and is mediated by the arrival of action potential at axon end, which is generated by voltage-gated channels. Synaptic vesicles are recycled locally to the axon terminus after fusion with the membrane. Depolarization of plasma membrane cannot by itself cause synaptic vesicles to fuse with the plasma membrane. For that matter, to activate the fusion of these vesicles with plasma membrane, an active potential has to be converted into chemical signal, and for this a localized rise in the cytosolic calcium concentration happens inside the presynaptic neuron cell. The transducers of the electrical signals are voltage-gated calcium channels, which are localized to the region of plasma membrane, which are adjacent to the synaptic vesicles. The action potential at axon terminus causes opening of these voltage-gated calcium channels that leads to the influx of calcium from the extracellular medium. Then there is the local calcium concentration near synaptic vesicle rises from 0.1 micromole, which is a characteristic of resting state, to up to a level of 1 to 100 micromole after arrival of this action potential. Calcium ions then bind to the protein that connects synaptic vesicles with plasma membrane and induces the exocytosis of neurotransmitter. After release of neurotransmitters, synaptic vesicles and other proteins which are involved in the process of exocytosis are retrieved by endocytosis in clathrin-coated vesicles. A released neurotransmitter resides in the synaptic cleft for short span of time, and after that it may be processed for enzymatic metabolization or bound to a postsynaptic receptor or sometimes reuptake by the presynaptic neurons even. When released neurotransmitter is taken by postsynaptic neuron, it is first detected by the receptor and it binds with the receptor of the presynaptic postsynaptic neurons. Neurotransmitter binding affect the postsynaptic neuron either as inhibitory or sometimes excitatory manner. The postsynaptic neuron may be connected to other neurons. Ultimately, it will create a new action potential at its axon hillock to release neurotransmitter to neighboring neurons. One can classify neurotransmitters on the basis of their structures. There are many different ways to classify neurotransmitters. However, they may be classified as conventional and gasotransmitters, which are generally called as unconventional neurotransmitters. The conventional may be subdivided into the small molecule neurotransmitters or neuropeptides, and these Small molecule neuropeptides are a different class of transmitters other than the amino acids, biogenic amines, biogenic amines, or sometimes the acetylcholine. Conventional neurotransmitters have certain common features as they are packed in synaptic vesicles, get released by elevated calcium ions, they can enter axon terminal and they can bind to the receptors on the membrane of postsynaptic cells. The conventional neurotransmitters are synthesized in the cytosol and transported for storage into synaptic vesicles within the axon terminals. These membrane-bound vesicles are near about 40 to 50 nanometer in diameter with a low lumen pH. Membrane of vesicles contain V-class proton pumps which are responsible for maintaining low lumen pH. Difference in concentration of proton gradient is responsible for import of neurotransmitter inside synaptic vesicles by ligand-specific proton-linked antiporters in vesicle membrane. In conventional neurotransmitter, small molecule neurotransmitters is a class in which you will find that amino acids neurotransmitters are present. These neurotransmitters include glutamate, aspartate, D-serine, gamma, aminobutyric acid, and glycine. If you see that gamma butyric acid is generally act as an inhibitory neurotransmitter that breaks the response of excitatory neurotransmitter that lead to relaxation. GABA is synthesized from glutamate by glutamic acid decarboxylase. Lacking of GABA in certain parts of brain may result in epilepsy. Anxiety disorders are also result of too little GABA production. The next one is the glutamate, which is an amino acid, is most common neurotransmitter in central nervous system, and it generally shows as an excitatory. In excess concentration, it is toxic to neurons. Next is glycine, which is also an amino acid. It acts as an inhibitory neurotransmitter in the brainstem and spinal cord, but it promotes the action of glutamate, hence also have excitatory functions within the central nervous system. It is sometimes synthesized from serine by enzyme serine hydroxyl methyltransferase. Next class is the biogenic amines. These neurotransmitters are made up from amino acid precursor through enzymatic action. This class of neurotransmitters include dopamine, norepinephrine, epinephrine, serotonin, and histamine. Norepinephrine and epinephrine are generally synthesized from tyrosine. Dopamine is a relative of epinephrine or norepinephrine having inhibitory action on nervous system. It is also synthesized from tyrosine. The concentration of dopamine is critical. Elevated levels in frontal lobes may result in mental disorders like schizophrenia, and limited amounts in the motor area of brain causes Parkinson's disease. The next biogenic amine is serotonin. It's a type of monoamine neurotransmitter and act as an inhibitory neurotransmitter for controlling emotion and mood changes. Major proportion of serotonin is produced by the intestine, approximately 90%, and is also present there, and the remainder is in the central nervous system. Too little amount of serotonin increase appetite for carbohydrate-rich food and sleeping, which ultimately results in the depression. It is a derivative of tryptophan, which is found in milk, and that is a reason why milk before bedtime help in sleeping. Cholinergic neurotransmitters are the derivatives of nucleotides such as adenosine. Adenosine has inhibitory effect on central nervous system. It has been studied when adenosine activity get decreased, the activity of dopamine and glutamate get increased. Now another class is of acetylcholine. It is the first neurotransmitter which was get identified. However, it has different structure as compared to structural categories of other conventional neurotransmitter. Acetylcholine acts as a transmitter at neuromuscular junctions connecting motor nerves to muscles. It activates skeletal muscles in the somatic nervous system and may either excite or inhibit internal organs in the autonomic system. Acetylcholine receptors blocking in muscle cells results paralysis. Acetylcholine also operates in many regions of brain with the help of nicotinic and nicotinic receptors. It has also noticed that the concentration of acetylcholine in brain get reduced to approximately 90% in Alzheimer patients. Acetylcholine is synthesized by acetyl coenzyme A and choline with the help of choline acetyltransferase enzyme. Acetylcholine is taken up by synaptic vesicles from the cytosol against a steep concentration gradient. The active transport is done with the help of an proton acetylcholine antiporter which is present in the vesicle membrane. It has been found that the gene encoding this antiporter is contained entirely within the first intron of choline acetyltransferase gene. Therefore, this is the example of evolutionary relationship for coordinated expression of two different but interlinked proteins. The next class is neuropeptides. These neurotransmitters are composed of at least 3 or more amino acids and hence are larger than small molecule neurotransmitters. The vesicle containing these peptides are larger than the vesicle containing small molecules and are correlated by calcium-dependent exocytosis with few differences. Many of these are co-released along with the small molecule transmitters. Endorphins are the example of neuropeptide. Endorphin is an endogenous morphine having structure similar to opioids. It acts as an inhibitory neurotransmitter that helps to reduce pain and leads to pleasure. The opioid drugs act by binding with the receptor of endorphin. Now coming to the next class, which is known as gasotransmitters, or sometimes we call it as unconventional neurotransmitters. This class contains nitric oxide, hydrogen sulfide, and carbon monoxide, etc. These molecules are not secreted by exocytosis means. These gaseous molecules are produced in the neural cytoplasm and immediately diffuse from cells into the extracellular fluid through membrane and finally into nearby cells to stimulate production of second messengers. Soluble gas neurotransmitters are difficult to study due to their rapid action and short span of existence, and they can exist up to the level of few seconds. Nitric oxide is one of the gasotransmitter which is highly reactive free radical, which is get synthesized by different forms of nitric oxide synthase. Nitric oxide has major function which includes vasodilation, penile erection, phagocytosis, and the formation of memories and learning. Next gasotransmitter is carbon monoxide. As we all know that carbon monoxide at higher concentration act as deadly poison. However, at low concentration, it have an important physiological role. It is secreted by heme oxygenase using heme from breakdown of hemoglobin. It modulates the function of cardiovascular system and also inhibit the aggregation and adhesion of blood platelets. Now coming to the classification of receptors of neurotransmitters. Neurotransmitter receptors are present in postsynaptic cells which receive signals and trigger an electrical signal through an ion channel activity. Binding of a neurotransmitter to a specific receptor cause opening of an ion channel which initiates the influx of ion and hence a change in the membrane potential of a neuron. Receptors of neurotransmitters are classified into two broad classes, which are number 1 ligand gated receptors and number 2 G protein coupled receptors, which are known as GPCRs. The downstream responses of each neurotransmitters are affected by their concentration and the strength of binding with these receptors. Ligand-gated ion channels are much faster as compared to G-protein coupled receptors. Ligand-gated ion channels are transmembrane proteins that are open or closed in response to the binding of a chemical or neurotransmitter and allow ions to pass through after binding with ligand. These channel proteins contain at least two domains, one for binding with the neurotransmitter and another to act as ion port. Nicotinic acetylcholine receptor is found at many nerve muscle synapses, which are known as neuromuscular junctions. This is the first ligand-gated ion channel which get purified, cloned and characterized at molecular level. The mechanism of neurotransmitter receptor provide a model for other neurotransmitter-gated ion channels. Activation of ligand-gated ion channels through acetylcholine, which induces transport of ion through these channels. If you see in the figure, you will find that firstly the acetylcholine binds with the receptor, then number 2 there is the opening of the channel, and the number 3 is the transport of ions into the cell. The nicotinic acetylcholine receptor is the example of ligand-gated ion channel which is present in membranes of muscle cells. This channel provides a passage for both sodium as well as potassium ion. The effect of acetylcholine on this receptor has been studied using patch clamp biophysical technique on isolated outside-out patch of muscle plasma membrane. Membrane potential measurement using patch clamp after binding of acetylcholine shows that these channels are capable of transporting 15,000 to 30,000 sodium or potassium ions in a millisecond. The resting potential of plasma membrane is near to equilibrium potential of potassium. Therefore, opening of these channels cause little increase in the efflux of potassium ions. Sodium ions come into the muscle cell due to the sodium ion electrochemical gradient. Therefore, the final outcome of these changes is the depolarization of membrane up to a minus 15 millivolt. If you see that the resting potential for membrane is minus 90 volt. This depolarization triggers opening of voltage-gated sodium channels that generate and conduct the action potential in the membrane of muscle cells. When depolarization reaches the T-tubules, which are special membrane invaginations, it causes the release of calcium ions from membrane to sarcoplasmic reticulum. Elevated concentration of calcium in cytoplasm induces muscle contraction, and because of this, signal transduction is induced by acetylcholine release from the single synaptic vesicle. Acetylcholine from single synaptic vesicle induces the opening of near about 3000 ion channels. Spontaneous depolarization results in the quantal molecules of acetylcholine, which further leads to opening of millions of ion channels. G-protein coupled receptors include huge family of surface receptors that activate trimeric G proteins. There are thousands of GPCRs found in humans which are responsible for visual, smell, taste, and maintenance of body metabolism. When a neurotransmitter or ligand bind with the G protein coupled receptor, a conformation change occurs in the GPCR and it acts as a guanine nucleotide exchange factor. The GPCR then activate an associated G protein, which is a heterotrimeric protein which have three subunits alpha, beta, and gamma. Inactive form of this protein have GDP bound, and all the three subunits are associated all together. The exchange of GDP with GTP in alpha subunit induces dissociation of beta and gamma subunit. Now, this GTP bound alpha subunit is a stimulator which further induces downstream signaling. The best known example of GPCR signaling is the binding of epinephrine or norepinephrine with beta androgenic receptors that leads to the activation of protein kinase A. If you see that beta androgenic receptors are 7 transmembrane proteins on binding of epinephrine, which leads to the conformation change in the receptor protein, which induces the exchange of GTP with GTP in a alpha subunit of this G protein. Activated G protein then bind with adenyl cyclase, which contain 12 membrane spanning helices. Binding induces catalytic activity of this enzyme, which produces cyclic AMP from ATP. Then cyclic AMP bind with 2 regulatory subunits of protein kinase A, which releases catalytic subunits, hence capable of phosphorylation of target proteins at serine and threonine. This whole cascade results in a number of other biological functions also. For example, release of glucose, which is required during fight or flight time from glycogen, and also stimulate the expression of certain genes by phosphorylation of transcriptional activators. A neuromuscular, or sometimes we call myoneural junction, is a chemical synapse which are formed by the contact of a motor neuron and a muscle fiber. Muscle contraction happens due to the transmission of nerve signal from a motor neuron to the muscle fiber at this neuromuscular junction. You will find in figure that how a muscle gets contracted with the help of neuromuscular junctions. Muscles get contract or relaxed when they receive signals from the nervous system. The neuromuscular junction is the site of the signal exchange. You will find that several steps are there which are required for this contraction. Number 1, the action potential reaches the axon terminal. Then number 2, you will find that voltage dependent calcium gates open, which allows calcium to enter the axon terminal. Then third, you will find that neurotransmitter vesicles fuses with the presynaptic membrane and acetylcholine is released into the synaptic cleft via exocytosis. Now, at number 4, you will find the acetylcholine binds to the post-synaptic receptors on the sarcolemma. And lastly, you will find that this binding causes ion channels to open and allow sodium ions to flow across the membrane into the muscle cell. This results the flow of sodium ions across the membrane into the muscle cell generate an action potential which travels to the myofibril and as a result in muscle contraction. Neuromuscular junctions are also associated with different genetic and autoimmune diseases. Junction muscular dystrophy is a genetic disorder which arises from mutated structural protein that comprises the neuromuscular junctions. Similarly, myasthenia gravis is an autoimmune disease which occurs when antibodies are produced against nicotinic acetylcholine receptor on the sarcolemma. So students, let's now summarize what we have learned from this module. You will find that a synapse is a junction formed between a neuron and another neuron or a effector cell. It is basically the space between neurons where the stimulation or inhibition of an action potential. You'll find that neurotransmitters are released by exocytosis a process in which neurotransmitter-filled synaptic vesicles fuses with the axonal membrane, releasing their content into the synaptic cleft. The exocytosis of neurotransmitters from synaptic vesicles involve a similar process of vesicle targeting and fusion events as seen in the intracellular protein trafficking. However, the secretion of a neurotransmitter occurs only at synapse and is mediated by the arrival of action potential at axon end, which is generally generated by voltage-gated ion channels. Synaptic vesicles are recycled locally to the axon terminus after fusion with the plasma membrane of presynaptic neurons. When released neurotransmitter is taken up by postsynaptic neuron, it is first detected by the receptor where it binds with the receptor, and then neurotransmitter binding affects the postsynaptic neuron either as inhibitory or excitatory manner. To activate vesicle fusion with the plasma membrane, an action potential has to be converted into chemical signal, and for this, a localized rise in the cytosolic calcium concentration happens inside the presynaptic neurons. The action potential at axon terminus causes opening of voltage-gated calcium channels that leads to the influx of calcium from the intracellular medium. Neurotransmitter may be classified as conventional and gasotransmitter or unconventional. The neurotransmitter may be further divided into structural bases into amino acid, biogenic amines, neuropeptides, etc. Conventional neurotransmitters have certain common features as they are packed in synaptic vesicles, get released by elevated calcium ions, enter axon terminal and bind to the receptor on the membrane of postsynaptic cells. These examples are glutamate, aspartate, or sometimes GABA and glycines. Acetylcholine is the first neurotransmitter which was identified. However, it has different structure as compared to structural categories of conventional neurotransmitter. It acts as a transmitter at neuromuscular junction connecting motor nerves to the muscles. It activates skeletal muscles in the somatic nervous system and may either excite or inhibit internal organs in the autonomic bodies. Receptors of neurotransmitter can be classified into ligand-gated channels or G-protein coupled receptors. A neuromuscular or myoneural junction is a chemical synapse formed by the contact of a motor neuron and the muscle fiber. Muscle contraction happens due to the transmission of nerve signal from the motor neuron to the muscle fiber at this neuromuscular junction. Nicotinic acetylcholine receptor is found at many nerve muscle synapses. This is the first ligand-gated ion channel which is get purified, cloned and characterized at molecular level. The mechanism of neurotransmitter receptor provide the model for other neurotransmitter-gated channels. Neuromuscular junctions are associated with different genetic and autoimmune diseases like Duchenne muscular dystrophy and myasthenia gravis, respectively. Thank you.